The purpose of the next scheme is to summarize some important points from the first days of the development of the zygote. So we know we got a zygote that still is surrounded by the zona pellucida, which is a glycoprotein envelope. Made of glycoproteins. This is the zygote. The differentiation potential of the cell is totipotent, because all the possible cell populations you may think about will develop from this cell. Now, during the first 20 to 24 hours, it undergoes its first mitotic division. As the daughter cells will still be kept inside the zona pellucida, which is kind of a rigid envelope, they need to be smaller than the mother cells. And these two cells are called blastomeres. So after approximately 1 day, we get a stage of 2 blastomeres. This process of the mitotic division is also called cleavage. During 48 hours after fertilization, the cells are undergoing second mitotic division, and still being trapped inside the zona pellucida. The outcome is 4 blastomeres. So it's a stage of 4 blastomeres. A day later, after fertilization, this third mitosis. So you get the stage of the cleavage results in a stage of 8 blastomeres. Of course, in reality, some would be hiding behind the other cells. So this is the stage of 8 blastomeres. Three days after fertilization, and at this stage, this early embryo starts to activate its own genes from the nucleus that has been formed during the formation of zygote. This important point is called embryonic genome activation, because since then the RNA is copied from the DNA of the zygote. Before, during all these first 2 days, the genes that have been translated were based on the RNA formed by the oocyte. So there is an acronym EGA, an important point in the human life where the individual starts to express his or her own genes. Now, during the 4th day, there's a breakdown of the zona pellucida. Of course, in the meantime, the number of blastomeres again increased from 8 to 16, then to 32, forming a solid object called Morula. So this inside is a solid object called Morula. Now the Morula will escape through an opening in the zona pellucida. There's a zona pellucida breakdown. And this escape is also called hatching. Like a small bird is hatching from the eggshell, from the shell, so it's a hatching. It makes possible for the Morula to grow and being transformed into an object that is not solid anymore but has a cavity inside. However, the cell populations will then differentiate into two different populations. An outer layer of cells that remains on the surface, that is called Trophoblast. And a mass of inner cells, the inner cell mass, also known as the embryoblast. The developmental fate of these two different cell populations will vary. So this is the inner cell mass, also known as the embryoblast. From the trophoblast, the extra-embryonic tissues like the placenta and so on will be formed. Within the embryoblast, two other vesicles, the contact of which will form the human body itself will differentiate. So trophoblast is the outer cells. There's a cavity here called blastocell. This is the spelling, and that's how we look like on the day 5 or day 6. Together, there's a hollow object. It's called the blastocyst. And the further fate of the blastocyst will be entering the uterine cavity, adhering to the mucosa of the uterus, and implant, which is called implantation or nidation. Implantation of the blastocyst.